Welcome to Medicalink. Don't forget to subscribe for more videos. Let's learn.
Hello friends, today we will be talking about glycolysis. So glycolysis, it is also known as EMP pathway. It is also known as EMP pathway. EMP pathway, and it takes place in cytoplasm. It takes place in cytoplasm of cell. This process of glycolysis takes place in every living cell. It takes place in every living cell. Even if some cells don't contain mitochondria. Even if some cells don't contain mitochondria, still we can't say that they are metabolically inert. They can generate their energy by means of glycolysis. So glycolysis is the first step in both aerobic and anaerobic respiration, and the process starts from glucose molecule, which is a 6-carbon compound. We should always know the starting point and the final resultant product. Here we have glucose as a starting point, and the final product is the pyruvic acid. So from one molecule of glucose we get 2 molecules of pyruvic acid. And also we should note that during the entire process of glycolysis there is no decarboxylation, that is, there is no generation of carbon dioxide molecule, and also oxygen is not used anywhere during the process, whether it is aerobic or anaerobic respiration. So the process of glycolysis, it starts from glucose molecule, and it is the first step in both aerobic and anaerobic respiration. So in the first step we have phosphorylation, where there is addition of phosphate group. So who will give the phosphate? Remember, whenever there is phosphorylation in a reaction, it occurs either by ATP or GTP. But in majority of the cases, it is ATP who will be giving the phosphate group. Here, ATP has 3 phosphate. It is adenine triphosphate. So, one phosphate will be given to the glucose, and glucose will be converted into glucose 6-phosphate by the enzyme hexokinase. And during this process, ATP will be converted into ADP by giving 1 phosphate to glucose. So you may ask that in glycolysis ATP is supposed to be generated, but here ATP is getting converted into ADP. It's because the reaction is investing ATP so that more ATP can be generated later on in this process. We see here that glucose is a 6-carbon compound. So it has 1, 2, 3, 4, 5 and 6 carbon, and it has a single side chain. When it gets converted into glucose 6-phosphate, 1 phosphate group will be added to the side chain of glucose. Now glucose 6-phosphate will undergo isomerization and will get converted into fructose 6-phosphate. Fructose 6-phosphate is also a 6-carbon compound. In glucose molecule there is only one side chain available where phosphorylation can occur, whereas in fructose there are 2 side chains. There are 2 side chains which are available for phosphorylation. So in this case, one side chain is free and can accommodate another phosphate group. So again ATP will donate its phosphate to fructose 6-phosphate and will get converted into fructose 1,6-biphosphate. So here fructose 6-phosphate will get converted into fructose 1,6-biphosphate in the presence of phosphofructokinase, where ATP will be converted into ADP by giving its one phosphate group to fructose 6-phosphate, so that the other chain will be fulfilled, so that the other chain will be occupied. Here fructose 1,6-biphosphate is also a 6-carbon compound. Now it has to be broken down. Now it breaks down into dihydroxyacetone phosphate. It breaks down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So this is a 6-carbon compound and it gets broken down into 3-carbon compounds. So dihydroxyacetone phosphate is also a 3-carbon compound and glyceraldehyde 3-phosphate is also a 3-carbon compound. Fructose 1,6-biphosphate gets converted into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate in the presence of the enzyme aldolase. This dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate and glyceraldehyde 3-phosphate can be converted into dihydroxyacetone phosphate as it is a reversible reaction, and this reaction will take place in the presence of the enzyme isomerase. Now we will take 2 molecules of glyceraldehyde 3-phosphate, which is a 3-carbon compound. In chemistry we have learnt that oxidation can be done by either adding oxygen or by removing hydrogen ion. So when we add oxygen or by removing hydrogen ion, oxidation process can occur. So in this case, hydrogen is being removed and this hydrogen ion is taken up by NAD plus and it gets converted into NADH and H plus. Since there are 2 molecules of glyceraldehyde 3-phosphate, so 2 molecules of NAD plus will give 2 molecules of NADH plus H plus ions. Glyceraldehyde 3-phosphate gets converted into 1,3-biphosphoglyceric acid. So where does this phosphate comes from? So during the process, inorganic phosphate is being added. So this is an oxidation process where 2 molecules of glyceraldehyde 3-phosphate gets converted into 2 molecules of 1,3-biphosphoglyceric acid in the presence of the enzyme dehydrogenase, where 2 atoms of hydrogen will be released and this hydrogen will be picked up by NAD plus and gets converted into NADH and H plus ion, and during the process there will be addition of inorganic phosphate. So we get 1,3-biphosphoglyceric acid. So how did we get this acid? We know that when hydrocarbons are oxidized, we get alcohol, and when alcohol is oxidized we get aldehydes or ketones, and when aldehydes and ketones are oxidized we get acids, and when acids are oxidized we get carbon dioxide. So glyceraldehyde 3-phosphate is an aldehyde and this is getting oxidized into an acid, that is 1,3-biphosphoglyceric acid, which is a 3-carbon compound. Now 2 molecules of 1,3-biphosphoglyceric acid gets converted into 3-phosphoglyceric acid in the presence of the enzyme phosphoglycerate kinase, where ADP will be converted into ATP. So now the ATPs are being generated. So in the initial phase ATP was being invested. So initially ATPs were being invested. Now ATPs are being produced. Now 3-phosphoglyceric acid will undergo isomerization in the presence of the enzyme mutase to get converted into 2-phosphoglyceric acid, which is also a 3-carbon compound. And now there will be enolation, where 2-phosphoglyceric acid will now get converted into phosphoenol pyruvate, that is PEP, in the presence of the enzyme enolase. And during this process, one molecule of water will be removed. So we get 2 molecules of phosphoenol pyruvate, which is also a 3-carbon compound. And finally, this phosphoenol pyruvate gets converted into pyruvic acid in the presence of the enzyme pyruvate kinase, where ATP will be released. So here ADP is converted into ATP. Since there are 2 molecules each, so 2 molecules of ADP will give 2 molecules of ATP, and our final resultant product is pyruvic acid. So we see that 1 molecule of glucose will be giving 2 molecules of pyruvic acid. And also we see that during the entire process, no carbon dioxide molecule was being formed. And also nowhere in the process oxygen was utilized. So it can occur in both aerobic and anaerobic respiration. Now coming to ATP calculation, we saw that number of ATP consumed was 2. 1 ATP is being consumed during the conversion of glucose to glucose 6-phosphate, and another ATP is being consumed during the conversion of fructose 6-phosphate to fructose 1,6-biphosphate. Okay. Now ATPs are produced when 1,3-biphosphoglyceric acid gets converted into 3-phosphoglyceric acid in the presence of enzyme phosphoglycerate kinase. Here 2 molecules of ATPs are produced. Also ATP is produced during the conversion of phosphoenol pyruvate to pyruvic acid in the presence of pyruvate kinase. So 2 molecules of ATPs are being formed here, and 2 molecules of ATPs are being formed here. So there are total 4 ATPs that are being produced at the substrate level. Now once pyruvic acid is formed, it can either go into aerobic respiration or anaerobic respiration. So in aerobic respiration, there is complete oxidation of pyruvic acid by means of Krebs cycle, and in anaerobic respiration there will be fermentation. So in aerobic respiration, this NADH plus H plus will form 3 ATPs. This will give 3 ATPs. So since there are 2 molecules, we will get 6 ATP. So during oxidative level, we get 2 molecules of NADH will give 3 ATP each, resulting in total 6 ATPs. So total number of ATP produced by substrate level is 4 ATP, and by oxidative level is 6 ATP. So we get total 10 ATPs. And out of 10 ATP, 2 ATP was consumed during this process. Therefore, net ATP generated during this process of glycolysis is 8 ATP in the aerobic condition, and in an anaerobic condition, ATP gained is total 2 ATPs, because in fermentation no ATP is generated. So total ATP in anaerobic glycolysis is 2 in number. So we see here that all the steps in these reactions are irreversible reactions, but there are 3 irreversible reactions occurring here. So first irreversible reaction is when it is getting converted from glucose to glucose 6-phosphate. Second irreversible reaction is when it is getting converted from fructose 6-phosphate to fructose 1,6-biphosphate. This is the second irreversible reaction. And third irreversible reaction is when phosphoenol pyruvate is getting converted into pyruvic acid. So this is third irreversible reaction. And this phosphofructokinase is a rate limiting enzyme, and this is the slowest reaction in the pathway, and it is a committed step as it converts fructose 6-phosphate to fructose 1,6-biphosphate. Fructose 1,6-biphosphate has no other fate but to complete the reaction. And coming to the regulation, so hexokinase are stimulated by insulin and it is inhibited by glucose 6-phosphate. Glucokinase or hexokinase is stimulated by insulin and inhibited by glucose 6-phosphate. Whereas phosphofructokinase, it is stimulated by insulin and inhibited by glucagon. This phosphofructokinase is also stimulated by AMP and fructose 2,6-biphosphate, and it is inhibited by ATP and citrate. Whereas pyruvate kinase is stimulated by insulin and it is inhibited by glucagon.